So if we look into the different genes, so Human Genome Project explored some interesting features about the human genes, which we can see statistics over here. Median is in stats, we see that it's a middlemost value, whereas a mean is the average. So if we look into the structures of introns and exons in proportion, we see that exons are relatively small and introns are large. So if we look into the internal exons, which are not the external but internal, so exons are separated by introns. If you keep it in your mind, the structure of a gene, so we have exons and then we have introns and then we have exons. So the internal exons are which are actually separated by the introns. So if you look into their size, so they are around 122 base pairs as far as the median is concerned, whereas the mean is 145. If we look into the introns, we see it's around 1 kb size as far as the median, and as far as the mean is concerned, it's around 3.3 kbs. So kind of there are some more values towards the right extreme. So normally the introns are larger in size. So the median is 7. So how many exons are present in genes? So the mean is 8.8. 3' UTRs are the untranslated regions towards the 3' side. So the DNA gets transcribed into messenger RNA, and then messenger RNA then translated. So UTRs are that part of messenger RNA which are not translated, but they are transcribed into messenger RNA. So if you look into 3' UTRs, they are around 400 base pairs. The median, I mean, is 770. 5' UTRs, 240 base pairs. 300 coding sequences, around 1 kb, 1.3 kb mean. So those coding sequences, then they get translated into the proteins. So obviously since we have a triplet codon, so there should be a ratio of 3:1. So 1/3 of this is roughly around 367 amino acids, and the mean amino acids coded in the proteins are 447. So genomic extent is 14 kb and 27 kb. Here in this diagram, we have the percentage of exons on the y-axis, and the x-axis are the exon lengths, and we are comparing 3 species. We are having humans, worms and flies. So if we look into the humans, there is a high peak where we have the size around 100 base pairs in length. So major proportion is where we have higher, around 6.5% of the exons are of this length, and then we have longer tail towards the right. If you look into the other species, they have their percentage of exons in different categories, especially in those higher categories, are relatively high as compared to the humans. So humans have relatively smaller size exons. Here is again the percentage of introns we see. So intron lengths are there. So there are different lengths. We are taking them 100 base pairs. Then we have 100 base pair to 2 kb region, and then 2 to 4 and so on. So if we look into the humans, which are represented by this black bars, so they are lower here. So mostly the human introns are of this length. So they are in this group. So they are around 101 base pair to 2 kb length. So mainly we have the longer introns in the humans. So if you look into the other species, especially the worms, so their intron sizes are smaller. And so there are more introns which are less than 100 base pair. Same way in flies also, but we see in humans introns are in longer length as compared to these 2 species. Here is the introns of small sizes. So their spread is seen over here. So percentage of introns again in these 3 species. So we have human, worm and fly. If we look into the humans, the intron length is, in terms of base pairs, is right here. So we are just looking into the smaller length introns. So the proportion of smaller length introns in humans is less as compared to worms and as compared to the flies. Human Genome Project also helped in cloning and discovery of different disease related genes. So topmost is BRCA, breast cancer susceptibility gene, and there are other genes which you can look into it. So obviously it helped explore the genes which are causing different diseases in humans. So what we have seen here is that human genome is having small exons which are separated by larger introns, and the proportion of the coding sequences in the human genome is relatively smaller.